Okay, thank you very much for this introduction and for this invitation to visit Vienna. It's always a pleasure. Well, this time I won't see the nice architecture and couldn't have beer and a restaurant with you, unfortunately. But it's still a pleasure, and I also thank you to have organized meetings with the students, even if it's with a video conference. Okay, so today I'm going to talk about hybridization, and I'm going to talk mainly about some populations of mussels we have recently discovered and that I think are really interesting. What happened now? Okay. I would first like to acknowledge my collaborators and mainly Alexis Simon, who was PhD student with me, and actually I'm going to present his work today, and even I'm going to use his slideshow. So thanks a lot to Alexis. And this work is also a collaboration with John Welsh from Cambridge for the theoretical part, Christelle Freys, that you may know because she was a postdoc for 3 years in Vienna at the IST with Nick Barton, and she's now back in France. Collaboration also with Frédéric Viard, who is working in Roscoff in Brittany, and she recently moved to Montpellier in my team. Hilde Schneemann, who is a PhD student with John, also doing some theory. And I'm also going to talk about some results we obtained on Australian mussel with a collaboration with Cynthia Reginos and her PhD student Eva Popovic, and also the contribution of all our team in Montpellier, biodiversity and evolution of marine species. So hybridization is quite widespread. It's widespread in agriculture. You are well aware that, for example, in maize, we are using inbred lines to benefit from heterosis. Here you have some cherry tomatoes, which are also F1 hybrids. They are very robust, very sweet, and it's actually the strain I am using in my organic garden. My children love these very sweet tomatoes. Hybridization is also used to create new varieties. For example, this strawberry has a taste of a pineapple. It's called a pineberry, and it's a F1 hybrid between a South American and a North American strain. Here you have a dog strain called Pomsky, very cute, which is a hybrid between a husky and a dwarf spit. Maybe you don't know, but it's also true in microbes, and I like this example in yeast. So the yeast used to make Lagers is actually a hybrid between Saccharomyces cerevisiae and Saccharomyces eubayanus. So usually I say maybe we can have a beer tonight, but today it would be a beer. It's also important hybridization in health and medicine. We have a recent example with coronavirus, SARS-CoV-2, which is a recombinant between a strain found in a bat and another strain which is related to a virus found in the pangolin. It's true in cancer, for example. In some leukemia, you have some cellular fusion that improve the ability of the cancer cells to propagate. You can also have some mitochondrial capture. Hybridization is important in conservation. Sometimes it's good, sometimes it's bad. it is bad when people don't want hybridization, then it's called gene pollution or extinction by hybridization. And sometimes it's good because the population is so deperish that hybridization is the only solution to rescue it. So you have a nice example in Florida panthers. Hybridization is also important in speciation research, and for a few decades now, you have very dynamic research in hybrid zones. So here you have my favorite species, Mytilus mussels, a very famous hybrid zone between Bombina toads, and here another hybrid zone studied in Vienna between two snapdragons species. And finally, you are aware that the human population is also the result of ancient hybridization between sapiens population from Africa that met Neanderthal and Denisovian population. It can explain some adaptation to altitude. So Tibetans adapted to altitude thanks to a Denisovan allele at EPAS1. And what is interesting is that Tibetan dogs adapted to altitude thanks to integration of Tibetan wolf alleles at the exact same gene, EPAS1. And another example of adaptive integration thanks to hybridization between Algerian mice and house mice that allowed house mice to resist anticoagulants thanks to a Spretus allele at this gene VKORC1. So hybridization is widespread, but we have very few knowledge of the expectation of hybrid fitness. Actually, we have only two simple determinisms that correspond to two extreme cases. We have a model of hybrid vigor that dates back to White's work, White's single locus theory. So the idea is that you have two inbred lines that have fixed deleterious alleles at different loci. And in the F1, every loci are at the heterozygous state. If deleterious alleles are recessive, you have a heterosis effect in the F1. In back crosses, you have only half of the alleles that are at the heterozygous state and the other half at the homozygous state. So you still have an advantage, a fitness advantage of back crosses between the two parental lines. At the other side of the divergence continuum, you expect hybrid breakdown, so fitness depression in hybrids, and the model that typically explain hybrid breakdown is Dobzhansky-Muller incompatibilities. So here you have the standard haploid pairwise Dobzhansky-Muller model. Species 1 has this genotype at two locus, little a at locus 1 and big B at locus 2. Species 2 has this genotype, big A allele at locus 1 and little b allele at locus 2. And you have two intermediate stages, little a, little b, and big A, big B. And this intermediate stage can be either fit if it's, for example, the ancestral genotype between the two derived genotypes, or it can be incompatible if you have negative interaction between alleles at the two derived alleles at the two loci. So divergence is important to understand hybridization, and the reason is that hybridization is the other side of the coin of speciation, and we know that speciation is a gradual mechanism that proceeds through intermediate stages during which hybridization is not completely interrupted and genomes remain permeable to genomic exchange. So here I show two examples of the effect of divergence. The very well-known work of Coyne and Orr on Drosophila flies, with a pair of fly species with increasing divergence, and you see that the success of hybrid crosses decrease with divergence. And here it's a work we have done using demographic reconstruction, and we showed using many pairs of species or population on a wide phylogenetic context that gene flow is possible when divergence is low, but when divergence is too high, gene flow is no longer possible. And you have an intermediate stage, often called the gray zone of speciation, where hybridization is possible, but some regions of the genome resist introgression or admixture, while some regions of the genome can be freely exchanged between incipient species. Okay, so to better understand and identify the genes involved in speciation or hybrid fitness depression, it's useful to use admixture mapping. So the principle of this approach is to take advantage of admixture between the migrant from a species into a recipient species. Then you have hybridization, recombination, and the two process, hybridization and recombination, create a diversity of genetic combinations between the two interacting genomes. And if you have several generations of mixing, you expect that post-diagotic selection sees genomic regions that resist introgression. And the idea is to find these genomic regions that stay pure or nearly pure for one parental ancestry. Here you have two nice examples in sword-tailed fish, so where these authors have mapped region of the genomes where you have a contrast with some fish with a high ancestry of parent 1 or the blue parent, and other fish with a high ancestry of the red parent. And here another example in sparrows. So in some Mediterranean islands, you have a genome that is highly admixed between house sparrows and Spanish sparrows, and they can identify region of the genome where the population is completely fixed for the house sparrow ancestry. Here, and you can even identify the genes in these regions that can be involved in species barriers. So the evolutionary biologists have this toolbox. We can use control crosses. In this case, we have only access of the first of the second generation of hybridization. So it's important because we can produce third generation hybrids that are not always easy to sample in the wild. These crosses are unfortunately rarely replicated. It's not that it's impossible, but it's often a hard work, and except in model system like flies or yeast, where you can replicate crosses, it's much more difficult for other species, non-model species. Often it takes one PhD project to do this, and people don't want to or don't get the funding to replicate crosses. The demographic context is very special because we force hybridization, and also we are dependent that crosses are done in the lab environment. We also have natural hybrid zones in secondary contact hybrid zones. They are usually post-glacial, so hybridization occurs for thousands of generations. We expect a stable state, so we have missed the initial stage of hybridization. Again, they are rarely replicated. The demographic context is badly known. We need to use demographic inferences to reconstruct the demographic context. It's difficult. But we are in the wild environment, so it's more the environmental conditions that matters for the species under consideration. But now, thanks or because of human activities, we can have recent secondary contacts because of introduction by human activities. In this case of recent secondary contacts, it's quite nice because we have only tens to thousands of generations, so we have access to the initial transitory phase that we are lacking in natural hybrid zones. They are often replicated because humans have introduced the same species at different places, so it provides a replication of admixture, so it's really nice. The demographic context, on the other hand, is often complex, or at least unusual, because you have a small propagule of introduced species inside large populations of resident species. So it creates a strong demographic asymmetry. And the environment is either the wild environment and sometimes a new environment, for example, a urban environment. So highly anthropozized environment. Okay, so for the rest of the talk, I will often describe the genotype with synthetic values. So you have the genome of a hybrid composed of chromosomal tract that comes from parent 1 or parent 2. So we can have the fraction of the parent 2, the ancestry of each lineages. It's often called the hybrid index. So it will be visualized by this kind of bars with a proportion of lineage 2 and proportion of lineage 1. And you can have also the proportion of the genome that is homozygous for parent 1, heterozygous, or homozygous for parent 2. So these values, the heterozygosity, P12, will be also used. So the hybrid index, H, is simply P2, the proportion of the genome homozygous for parent 2, plus half P12, so the proportion of the genome that is heterozygous. So the possible outcomes of a secondary contact are diverse. You can have a coexistence of the two species with no or very few hybridization. You can have invasion of one species and the other disappear. Maybe you can have the trace of this invasion with a little ancestry. of the local species in the genome of the invited species. So a nice example is the presence of Neanderthal ancestry in European and Asian population in humans. You can have the formation of a hybrid zone and integration through these hybrid zones in the parental population. So integration will be stronger close to the hybrid zone and less important in distant population far away from the hybrid zone. And finally, you can have homogeneous admixture, where you have a balanced ancestry of the two parental population in an admixed population. And so these outcomes are modulated by many factors. First, the level of divergence, as I began to explain. The longer was the divergence, the more barrier to gene flow can have accumulated in genomes. It also depends on this number of species barriers, but also their genetic determinism, their genetic architecture, how barrier loci interact in the genome. It also depends on the fitness of the parents and also their hybrids, but often we undermine the importance of parental fitness. But it became clear to me that parental fitness is important when we study hybridization, and it's also modulated by demography. If the two genomes in contact are in balance proportion, or if you have one parental genome that is majoritary and the other is minority. Okay, so this was a quick overall introduction. I now will present my study system, the Mytilus species complex. So marine mussels, actually the one you eat with French fries, for example. So in the northern hemisphere, three species are described: Mytilus truculus, Mytilus edulis, and Mytilus galloprovincialis. Mytilus galloprovincialis is only found in Europe, in the Mediterranean Sea, and in the Atlantic Ocean. Mytilus edulis is found on the both sides of the Atlantic Ocean in Europe and North America. And Mytilus truculus is found in the Pacific Ocean and in Northern America and the Atlantic coast, and also in Europe in the Baltic Sea. So these species are in contact and form hybrid zones, and each time you have hybridization and integration going on through the hybrid zones. We can also describe lineages within species. So for example, in Mytilus edulis, you have a lineage in the southern Europe. Maybe let's look at this zoom here on Europe. So it's the dark blue, and you have a hybrid zone in Denmark with northern lineage that is found in Norway and Scandinavia. And actually, this Northern Europe lineage is intermediate between Southern Europe and America. And in America, you have the American lineage. And for Mytilus galloprovincialis, you have a Mediterranean lineage and an Atlantic lineage with a hybrid zone at the entrance of the Mediterranean Sea here in Algeria. And you also have a genetic differentiation between Baltic truculus and other truculus. So we know that these contact zones are secondary contact, and We know that the divergence between lineages are quite old. So, for example, between Trosulus and the other species, it's around 3 to 4 million years. Between Edulis and galloprovincialis, it's around 2 million years. And for lineages within species, it's a few glacial oscillations old, so a few 100,000 years. We know that the differentiation is highly heterogeneous in the genome. So here you have the FST distribution between Edulis and Gallo. So you have regions that are highly differentiated between the two species and some regions that are very lightly differentiated. So it's in accordance with a semi-permeable species barrier with genome regions that resist hybridization and integration, and genome regions that can be exchanged nearly freely between species. We know that it's secondary contact thanks to demographic reconstruction, and probably it's a periodic secondary contact with isolation between lineages during glacial periods and contact between interglacial periods. And what we have done is to select SNPs that are the most differentiated between lineages to create a small number of SNP markers that are highly informative about the ancestry of the individuals that we can use on many individuals. So we focus with this panel of ancestry informative SNPs on a region of the genome that we think resist integration. So this panel is quite small, it's only 100 SNPs, but we genotype 10,000 individuals. So if we focus on reference populations, the panel works very well. Here on this PCA, you have Mytilus trosulus, here Mytilus edulis, and Mytilus galloprovincialis. On PC3, you have the differentiation between the Mediterranean lineage of galloprovincialis and the Atlantic lineage. And on PC4, the differentiation between Edulis lineages with America, north of Europe, and south of Europe. So this is when we focus on reference population, so far from hybrid zones. If I can come back to the first factorial plan, I can add the 10,000 individuals in contact zone. And what you can see is that the genetic gradient is fully filled by these individuals. So you have really a gradient of hybrid genotypes between the parental species. It's also true between lineages within species. So you can see that even if we focus on the most differentiated region of the genome, so regions that should not be exchanged and should resist hybridization and integration, we find, when we sample in contact zones, we find all these gradients of intermediate genotypes. Just briefly, we know the biology behind these species barriers, the mechanism of reproductive isolation. We have found that there is asynchronous reproduction. The two species often do not reproduce at the same period of the year. You have assortative mating. So gametes can choose each other, and you have an excess of ultra-specific fertilization when compared to heterospecific fertilization. You have habitat specialization. I won't going to talk a lot about this today because I prefer to focus on this mechanism, hybrid fitness depression. That is the only mechanism that can really work on a large fraction of the genome and can really explain the many barrier loci found in the genome. But remember that other mechanisms are also at play in this system. Okay. So one of the species from the northern hemisphere, Mytilus galloprovincialis, has become an invasive species. It has been introduced and invaded Asia, Western North America, South America, and Australia and New Zealand. I should say that in the southern hemisphere, you have 3 other species: Mytilus silensis in Chile, Mytilus platensis in Argentina, and Mytilus planulatus in Australia and New Zealand. So that's the species status, but actually planulatus is very related to galloprovincialis. It's probably a trans-equatorial migration during the last interglacial, and platensis is well related to Mytilus edulis. Again, it's probably trans-equatorial migration during the previous or 2 previous interglacial. Mytilus chilensis, on the other hand, is quite divergent from the other species. But what is important is all these introductions of Mytilus galloprovincialis that each time hybridize with the local population, except in South Africa because there were no Mytilus mussel before the introduction of Mytilus galloprovincialis. So the introduction is due to human activities, either shipment or aquaculture activity. And you also have introduction at a smaller scale. For example, in Northern Europe, population of galloprovincialis has been found in Norway, in Svalbard, or in Iceland. And I'm going to talk about this population in France soon. So in Australia, and the introduction of galloprovincialis has been followed by a strong admixture. It's a paper by Eva in evolutionary applications. You have this admixture between planulatus, that is the native species, and it has been admixed either with the Mediterranean gallo or with the Atlantic gallo at 2 different harbors in Australia. So you see that the admixture is quite extensive in this case. On the other hand, in California, galloprovincialis has met Trosulus, which is much more divergent. And in this case, you rather have coexistence of the 2 species with very, very rare hybridization, and mainly F1 hybrids with a very, very small signal of introgression. Okay, so with the Mytilus model, we have we can benefit from a control process, from natural hybrid zones, and now we have recent secondary contacts. And we can compare how admixture and introgression proceed in these 3 situations. So I'm first going to explain how we discovered newly admixed population in ports, then how we tried to map strong deviation in the genomes of admixed populations. And finally, I will explain briefly a polygenic model that can explain the unexpected result we obtained. So first, the discovery of new anthropogenic admixture in mussels. So we began to sample ports because actually we were tracking for the introduction of Mytilus trosulus. We thought that Mytilus trosulus could be found in France, but it's not what we found. We found something else. So here you have the population, the natural population in France, Mytilus edulis, hybridization in southern Brittany, Mytilus galloprovincialis in Brittany, another hybridization in Normandy, and then edulis again in the British Channel. And we have sampled in 5 ports. So it's this kind of ports, big commercial ports with a container ship and a very strong activity, a strongly urbanized environment. And what we found is that all the mussels found in these ports, we call them dock mussels, have an intermediate composition between edulis and galloprovincialis, but not with the Atlantic lineage of galloprovincialis, which is the galloprovincialis lineage found in the wild at this place, but with the Mediterranean galloprovincialis. So our interpretation is that Mytilus galloprovincialis from the Mediterranean Sea, has been introduced in these ports and hybridized with Mytilus edulis, forming this admixed lineage we call duck mussels. And here you have the hybrids found in natural hybrid zones that are, as expected, found between edulis and the Atlantic gallo. So to study the duck mussels, we use 2 genotyping strategies. First, our panel of 100 ancestry informative SNPs, and then we wanted to sequence the whole genome, so we used low coverage whole genome sequencing. Just a detail, the mussel genome is quite big. It's 1.5 gigabase, and it's very complex, so it's quite a difficult genome to work with. And with a lot of diversity, the nucleotide diversity of mussels is a few percent, between 4 and 7%, and the divergence between species is between 2 and 4-5%. So we have to deal with a very divergent sequence in the process of mapping reads and calling SNPs. Okay. So what we did was first to date the admixture of duck mussels. So either with a small panel of markers, 30 markers, but that we managed to place in a genetic map. So we have the recombination between some of them. Maybe it looks like a small data set, but actually it's a very good data set to use the recombination that has been inferred in crosses and not deduced from population genetic data. And we estimate that duck mussels admixed with, Mediterranean gallo admixed with edulis between 4 and 14 generations in the past, so it's quite a recent admixture. And with genomes, we use a method, ancestry HMM to date the admixture, and you have the results for the ports. So the estimation is quite older, but still recent, between 20 and 80 generations. So, but the problem with this method is that it makes a number of assumptions. For example, that the variation is neutral, and it's far to be the case when you admix mussel species that are 2% divergent. And it also assumes that you have no migration from the native population, and it's also likely false. And so you end with the surprising result that the estimation for a sample in the port of Cherbourg from 2017 is estimated to be younger than a sample from 2013. So you have 15 years between these two sampling time, and so you would expect this date to be 15 years more recent, and you have the reverse. So I think this result should be taken as evidence that it's very difficult to date admixture, and that the estimations should not be taken at face value. But at any rate, we estimate recent admixture, and we expect that these ports that have been built in the 19th century are quite recent. So we are dealing with an admixture that we know is quite recent. Okay, so where are these duck mussels? And it's quite surprising to me. They have been found only in ports. So here you have the port of Le Havre. So you have some basins, the basins, and all the duck mussels are found in these basins. And as soon as you go outside of the port, so here you have a sample on the other side of the port, and you only find the native mussel, Mytilus edulis. You also see that the admixture proportion in duck mussels are quite homogeneous, roughly 70% galloprovincialis, 30% edulis, with a low variance between individuals. And you have a zone of coexistence between the duck mussels and the native edulis at the entrance of the port. Here you have the other port with quite the same result. In Cherbourg, all the duck mussels are really in the port. And when you go outside of the port, you find the native M. edulis populations. The same in the port of Saint-Nazaire. Here in Saint-Malo, the native species is actually the Atlantic lineage of galloprovincialis. But again, the duck mussels are found only in the port. And in Brest, it's the only place where the duck mussels, the hybrid lineage, managed to go outside of the port and to colonize this estuary, which is named the Elorn estuary here. And also in Brest, the native species is not edulis, but is Atlantic gallo, and you have a beginning of admixture between the duck mussels, so that are hybrid lineage, with the Atlantic gallo. So you create hybrids that have three ancestries, Mediterranean gallo, Atlantic gallo, and edulis ancestries. So I talked to you that we also have found some introduced gallo in Norway, in Northern Europe, but in this case, they were admixed between edulis and this time the Atlantic lineage of gallo. So they are not like the duck mussel. They are an independent admixture, but with the other lineage of gallo. And it can be seen here on the PC4 that the edulis background, the edulis genome that is admixed with the galloprovincialis genome is coherent with the local lineage found in Norway, which are the edulis from Northern Europe. So just here to show you where they are, here in the Lofoten Island, here in the southern of Norway. Okay, so to sum up, in this system, we have blood crosses between edulis and Mediterranean gallo. We have 5 replicates of admixed mussels between, again, edulis and Mediterranean gallo in ports. We have natural hybrid zones between Atlantic gallo and edulis, and admixture in Norway between Atlantic gallo and the northern lineage of edulis. And we have 2 replicates of this admixture, and we can also have 3 replicates of natural admixture in France and in Scotland and in southern England. So we wanted to map loci with strong deviations from expected ancestry. So what is the expectation? Imagine that the determinism is under dominance. The expectation is very simple. You start from an admixed population and you expect the major parent allele to fix because of under dominance. And the process is very quick. It takes a few tens of generations to fix. So even in duck mussel, we expect to be close to fixation. The other model is the Bateson-Dobzhansky-Muller incompatibilities. So you have the parental genotype and all the hybrid genotype. One corresponds to the ancestral genotype in green with ancestral alleles, and all the combination with derived alleles at the 2 loci result in a lower fitness. Okay, so you see here in recombinant hybrids, you expect a lower fitness under this model. Okay, and with this model, you again expect fixation of the ancestral allele of the major parent, the parent that is more frequent at the time of admixture, at one locus, and once the ancestral allele is fixed, you maintain polymorphism at the other locus, because the big B allele is no longer in presence of the big A allele at locus 1. So under dominance of simple pairwise Dobzhansky-Muller incompatibility, you expect fixation of the ancestral allele of the major parent, at least at some loci. So what we expect is, if we start with an admixture, so intermediate allele frequencies at loci, we expect, you expect the frequency of the major parent allele to increase and reach fixation quickly, and it will happen in a region of the genome that are a barrier to gene flows, resist introgression. Okay, but it's not what we observe. I must say that I was very surprised. If we use our ancestry informative SNP, we have no fixation. So it's a surprise because we really, I must say again, that we selected this SNP to be the most differentiated, so they are fixed or nearly fixed in gallo and in edulis. And you have here the allele frequency in crosses, F2 back crosses, and in ports here. So if you are not surprised, I can show you the result with a genome sequence. So the sample size is not as large. So here we have a few fixations in some samples. But if we mixed all the port samples, you can see that we don't have any fixation. So even if we sequence the whole genome, we have no fixation in the genome. So the question is that maybe it's simple neutral admixture. So how can we prove that we still have a selection? And the idea is to correlate deviation in allele frequencies between samples. So here you have three loci, the red locus, the green locus, and the blue locus. You have the expectation knowing the admixture proportion in population A or in population B, and you can compute the deviation observed in population A or in population B. So in this example, the red locus shows strong deviation toward the gallo allele in the two populations. The blue locus, a strong deviation towards the edulis allele in the two populations, and the green allele does not deviate. And if you do that, we obtain very nice correlation between populations. If we correlate ports, for example, duck mussels, we have a very nice correlation of deviation between ports. So the same loci with an higher than expected frequency of gallo are the same, and a deficit of gallo ancestry are the same also. If we compare natural hybrid zone, again we have a nice correlation. So we can have the hypothesis that maybe it's not independent replicate. You can have admixture in one port, and then this admixed mussel have been introduced in another port. So they have a shared history, and this is the reason why you have correlation. But what is nice is that we still have good correlation. For example, if we compare admixture in Norway, so introduce gallo in Norway, that admixed with northern edulis, and admixture in Aberdeen, for example, in Scotland, which we know is an independent admixture because the native edulis background is different. It's Southern Europe edulis background. Here you have the correlation between duck mussel in port and back crosses in the lab. So back crosses in the lab are obviously independent replicate, and we obtain this nice correlation. Locus that deviate toward gallo are the same in back crosses in the lab and in port mussels. But sometimes the correlation is not significant, or even sometimes negative, and it's the case when the galloprovincialis background is not the same. When we compare admixture that involve Mediterranean gallo and admixture that involve Atlantic gallo. And if we use genome, I don't show it here because they are not as nice because sample size are much smaller, but we still have a very strong correlation if we compare ports, duck mussels, very nice correlation if we compare natural admixture at several places, or if we compare natural admixture and Norway admixture, we still have a correlation. But when we compare admixture with that involve Atlantic gallo and Mediterranean gallo, the correlation is not so strong and non-significant. Okay. And briefly, we also, with Eva and Cynthia, we analyze admixture in Australia, so between Planulatus and Atlantic gallo or Mediterranean gallo, And we also found correlation in the deviation. So you see that this pattern is replicated many times. So now, I don't know what is the time actually. Okay, I'm long. So briefly, I explain a polygenic model. So the starting point is that in crosses, forget this because I'm too slow. When we studied the segregation distortion with 33 markers in F2 crosses, we were surprised that we had a slight advantage for galloprovincialis genotypes, an advantage for multi-heterozygous genotypes, but we did not find strong mono or bilocus deviation. To explain this, we use a fitness landscape model. So the idea is that we have a chain of substitution that link the two parents, parent 1 and parent 2. So here you have 7 substitutions, 7 mutations. These mutations have a phenotypic effect. So we have phenotype during this chain of evolution. And we use a fitness landscape, and the fitness landscape is the Fisher geometric model. What is nice with the Fisher geometric model is that a mutation, so a mutation is a vector in the fitness in the phenotypic space, a vector like this. In this case, you see that the mutation is deleterious. The fitness is higher at the beginning than at the end of the vector, but the exact same mutation can be neutral. It does not change the fitness, and the same mutation can be beneficial. It increases the fitness. So you have the spirit of the Dobzhansky-Muller model where the fitness effect of one mutation depends on the genetic background in which it is found. And to characterize hybrid genotype, we use these two parameters, P12, heterozygosity, and H, the hybrid index. So we can start from the ancestor, and you have a chain of mutations that go to parent 1 with some position on the genome. You have the chain that go from the ancestor to parent 2 with other position in the genome. And what you do to create an hybrid is simply to draw at random the mutations from the two genomes. And you obtain the phenotype of the hybrid, and with the fitness function, the fitness of the hybrid. So here, for example, this hybrid, the H, the hybrid index is 4 over 7. So this is a haploid model, and it's easy to create a diploid model. Here it's a case with additivity, so simply the two alleles in diploid genotypes add additively on the phenotype. And recently, in this paper by Hilde, we introduced phenotypic dominance, so we can even introduce phenotypic dominance. And it's the same to obtain the hybrid genotype. You randomly sample mutation in the two diploid genome and obtain a diploid hybrid. This time that is depicted by H and also heterozygosity P12. If we come back on the chain of mutation, we can show the value of the phenotypic traits during evolution. What happens when the chain goes through the common ancestor between the two parents. So this chain has happened at high fitness value because it's really what happened during evolution. But when you create hybrids, you draw this mutation at random, and what happens is that it can go in very weird direction because it has not been constrained by evolution. The this chain has not been constrained by evolution. And what is very nice, it has been shown by John Welch, is that this work between P1 and P2 can be described by a Brownian bridge interpolation, which is a Brownian motion that is constrained at the start to be the phenotype of P1, and the end of the chain, which is the phenotype value of P2. Okay, to be brief, we can obtain the fitness of the hybrid, but for simplicity, we can use a scaled value, which we call the scaled breakdown score, and actually it's simply the distance between the optimum of the fitness landscape and the hybrid genotype, so RHO. And this value, the phenotype, I would say, of the hybrid, can be described by 4 components that can be understood with distance in the fitness. So you can understand hybrid in a geometric fashion. So the first term is the average distance between the two, between parent 1 and the optimum, and parent 1 and the optimum. It measures how far the parents are on average from the optimum. And so it says that the fitter are the 2 parents on average, the fitter are the hybrids. The second component involves the distance between parent 1 and the optimum and parent 2 and the optimum. So these 2 distances here, R1O and R2O. Sorry. And so this component measures how this component shows that hybrids are fitter with more alleles from the fitter parent. So it explains the asymmetry between the 2 genomes in favor of the fitter parents. The first component is simply an intrinsic component that is heterozygous, an intrinsic benefit of heterozygosity, so P12. And the last component is the intrinsic effect of admixture. So these 2 components are completely independent of the optimum. Here it's just P12, and here what is involved in this component is R12, so it's the distance between the 2 parents. It's completely independent of the optimum. And what this component says is that this intrinsic effect of admixture is a cost when this distance is below 4, and is a benefit when this distance is higher than 4. And what is important to understand is that if these 2 components allow a nice decomposition of the expected phenotype of the hybrid, they are constrained by each other. For example, it's very difficult to have a long distance between P1 and P2 if the 2 parents are very close to the optimum. So if the 2 parents are very close to the optimum, R12 can only be small. So for R12 to be large, the 2 parents need to be maladapted. Only in this situation, we can have a large R12, and so a benefit of admixture. And so R12 tells us whether parental phenotypes have diverged more or less than would be predicted by a random walk model of evolution. And so it tells us about how was the mode of divergence. Imagine that we have this kind of divergence with adaptation to 2 different optimum in P1 and in P2. In this case, you can see that the walk is much more straight than would be expected if it was a random walk, and R12 is in this case square R12 is higher than 4. Here you have the random walk, so it's really a divergence by simple drift, and in this case, R12 square is near 4. If you have a system drift, so in this case, after the adaptation, the population drift around the optimum, so it's like nearly neutral evolution around the optimum. So it accumulates many mutations in many directions, but at the same time being staying fit and adapted to the optimum. In this case, R12 square will be below 4, or if you have a complex adaptive change that go in any direction, again, R12 square will be below 4. So at the end of the day, R12 decreases over time due either to system drift or complex environmental change. So if divergence is long enough, you will always converge to an intrinsic cost of admixture. Okay. And we believe this is the case in mussels. The two parents are well adapted, and in any case, have a very long history of divergence. And in this case, this is the kind of fitness landscape we expect in the space defined by the inbreed index and heterozygosity. So genotypes are fit on the border of this triangle and unfit in the center of this triangle. And so if we compare backcross and F2s, we expect fitness decrease only in the F2s, and it was what we observed actually. We observed hybrid depression only in the F2s but not in the backcrosses. And in the F2s, we expect an advantage of heterozygous genotypes, and it's actually what we observe. We have an excess of heterozygosity in F2 that is not observed in the backcrosses. So the model explains quite well what we have observed in lab crosses. However, it's still possible that Mytilus galloprovincialis can be a little fitter than Edulis. So in this case, the fitness landscape is like this. And in this case, we have an advantage toward galloprovincialis genotype that can explain actually the genotype composition observed in ports. Maybe the admixture proportion were more balanced at the beginning of the admixture, and the whole composition of the genome have moved toward galloprovincialis ancestry. Okay, so it's time to conclude. Sorry if I am a little bit long. So we have quite a nice panel of tools in Mytilus to study hybridization, lab crosses, anthropogenic secondary contacts, and ancient hybrid zones. We have shown that the polygenic model can well predict hybrid fitness in lab crosses. And in anthropogenic admixture, we have two different events of admixture in duck mussel and in Norway, and they correspond to replicate either of lab crosses for duck mussels or for natural hybrid zones for Norway. So we began this work with one expectation that we have barrier loci in the genome that resist a lot introgression, that can be simple Dobzhansky-Muller incompatibilities, and then that we will observe fixation in admixed mussels, and it's actually not what we observed. We observed that duck mussels perform quite well despite a strongly homogeneous admixture all along the genome. And so the view has quite changed. We shift from a genic view of speciation with strong effect barrier regions, also called speciation genes, to a more polygenic view like this, where the effects of genomic regions are modulated by their content, the number of genes, the density of genes, and the genetic landscapes, for example, the recombination rate. And all this modulates finally how the two genomes interact in admixed population. So with many loci everywhere in the genome. Okay, thank you.